In January of 1993, Arecibo received a message from an extraterrestrial, unknown source.
That's great.
Intelligent life beyond this planet.
Yes. Turned out to be a new sequence of DNA with a rather friendly instructions on how to combine it with ours.
The scientific premise behind the movie Species was really an interesting one. The idea of whether or not one could fuse two very dissimilar sets of genetic instructions into a single entity. It would entail really a phenomenally detailed understanding of our biology to do that. Because not only would we have to understand our biology, but also what other sets of instructions could be interlaced and interleaved with our own.
It's 100% fiction, but in some respects, it's 100% fiction because you're taking the genetic material from a creature which is very, 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 very far removed from a human being, theoretically. If you're taking something from an alien which has been on another planet or another solar system, they would have had their line, millions and millions of years of evolution in which their genetic material would be very, very different from our genetic material. And so therefore going from that distantly related creature to human beings might not even work.
The human genome is made up of 35,000 genes. The number of genes that we have and the kinds of genes we have are not much different from a lot of other organisms. A classic example people talk about are chimpanzees and things like that. The variations is in a few percent. Well, why do you think we're so different from these other organisms? It's not because we have one incredible genius gene. It's because it's the way we organize our genes. It's the way we put them together, when they're turned on, in what time of your life, in what physical display in your brain. That is the core reason why we're so different from other organisms.
I felt it.
What?
Life.
People understand about male and female mating. Male and females mate, each one presents 50% of their genome, and you get a new offspring. That's 50%. If you are talking about taking 50% of human DNA and 50% of another organism's DNA and coming up with a new species, I would say that's an incredible challenge. It's an incredible challenge, not likely to be doable immediately. I mean, it would take a lot of time, a lot of effort, and many years.
Genes are the instruction or blueprint for basically how an animal works. You know, from our skin to our eyes, everything is coded in DNA and similar molecules. When we put DNA, new DNA that's never been there into an organism, we don't know what other genes it's going to turn off or turn on, and so there's always unexpected things that happen.
Genetic engineering is a way that you can manipulate genes, and so you can change genes, and you can do that by altering the DNA in some very sophisticated and highly specified ways. So you can do it on a base-by-base basis. In other words, change the 4 letters of the code A, T, C, or G in a highly specified way. You can do it at any place on the DNA that you'd like to do it, or on a much broader basis, you can move things around in the DNA where you're swapping genetic information from intra-organism in between its own organism, or you can move DNA around between organisms. And so we can think about that as engineering a gene.
The entire genome is a very complex book, and to make a change here or a change there, it's really hard to tell what the outcome is going to be. The variability be extremely high. We don't really understand how the genome comes together to give us the information that it takes to make an animal, for instance, or to make a part of an animal, or to make a piece of a cell. So to make a small change could have random, unexpected consequences.
What we're able to do now is put a few genes into plants or animals. So you have these complex interactions between genes, and most likely if you put a lot of different genes, they're going to interact in ways that cause the death or major deformity of either a human or an animal. With humans, again, it's completely speculative. But there are well-established theories in population genetics and in molecular biology where we know that if you have a gene and the proteins that they make, which do the work of the cell, and the genes code for, have the instructions to make the proteins, and then the proteins do the work of the cell, those proteins often interact with each other. And they have to interact in a very precise way. So if you have genes from one organism and put it in another organism, many of those genes are likely to have enough similarity that they will interact in some ways, but they won't do it in the right way.
In terms of the basic technology, we have the technology today in small part, maybe not in the magnitude required to construct an entire alien entity. For example, a few years ago, scientists synthesized a poliovirus. Now, poliovirus is pretty small and not very complicated compared to us. The genome, the genes of a poliovirus that are written in its DNA are only about 6,000 letters long, right? The 4 letters in DNA, A, G, C, and T. And that 6,000 letter long piece of DNA has been synthesized and put into cells and created polioviruses. In principle, we have all the technology we need to create organisms from scratch. Now, that said, going from an in-principle ability to do something to actually being able to do it's really a far cry. The human genome, for instance, is 3 billion letters long, and the poliovirus is 6,000 letters long. So it's going to take quite a while before we can get up to something as complicated as the human genome.
It is possible that, we might be able to take species that are closely related and try mixing, those types of organisms together. But there's still a lot of information, a lot of work needs to be done on figuring out the human genome, let alone the genomes of other organisms that you're trying to mix things with, or the genomes of 2 different species have to be well mapped out and understood first.
Let's say in the case of what we do now, to again take an egg, take the nucleus out of that egg, and then put a nucleus back into that egg, not from that individual, let's say, not from that person, but from another individual of exactly the same genus and species, of the same organism. That's commonly done in this day and age to try and establish clones of these animals. And doing that cross-species is something which is, in some respects, borders on the unethical, because you don't know exactly what kind of animal you'll get out. Let's say taking the egg of a mouse and putting the genes or the nucleus of, let's say, a cow in there.
So with regard to the ethics of molecular biology, there's 2 issues. One is, what are the ethical issues? What are the ethics associated with this? If we can modify organisms, including even ourselves, that's fine. That's a different question from whether or not we should. A separate issue, though, is once you've defined a set of ethics, how do you enforce them? What happens, though, when this becomes so easy that some high school student can do it as a school project, or some amateur scientist can do it in his garage after work?
Most of the people who generated the biotech, the whole recombinant DNA, the cloning of pieces of DNA, or even who've done the cloning of organisms like Dolly and these other things, they're motivated by one of 2 things. They're motivated either by just trying to understand the biology, period, or they're trying to do it for practical purposes. Human embryonic stem cells is an example. You're trying to help people in some therapeutic application. What society decides to do with that is a societal question. It's always a tough one for when scientists are asked, how far do you want to go with a particular technology? The people who developed atomic energy, they weren't advocates of doing the nuclear, the atomic bombs. I mean, they just developed a technology. Other people make decisions about where you want to go with that. It's the same thing with cloning. There's no question that being able to manipulate DNA, taking your DNA and putting it into an organism to propagate a particular gene or something like that, is useful. That's the basis of a huge part of our biotech industry. But how far do you want to go with that? Do you want to make clones of yourself? Do you want to hybridize your DNA with an alien's DNA, for example? Those are questions that society has to answer, because the scientists can make it happen. It's a question of do we want to have it happen from a societal point of view.
She would make one excellent biological weapon. If someone out there thought we were some kind of galactic weed that was going to spread through the universe.
So what you're saying is that she's the cure with the disease.
It's well said.
Genetic engineering is a very, very old science. In fact, we'd have none of the foods that we eat in the grocery store without genetic engineering, and you'd have none of the meat that you would eat without genetic engineering, because all of the animals that you eat, all the domesticated animals, have been genetically engineered over tens of thousands of years. And every single plant that you buy for vegetables in the grocery store, whether it's corn or broccoli or cauliflower or cabbage or strawberries, any of these things, or carrots, have been genetically engineered. And what that means is that from a classical point of view, people have taken wild organisms, whether animal or plants, that just grow naturally in the wild, and they've selected it for a particular trait. Let's say in the case of corn, the original corn had a cob that was about this big. And so people looked at mutations that just occurred naturally, and maybe someone about 10,000 years ago saw that there was a cob that was a little bigger, and they said, oh, well, that's really useful, you know, it's got more seeds, you know, I'd like to use that. So they put it aside, and then, because in the case of plants, they can do it with themselves, so to speak, they're able to perpetuate that and then keep selecting for bigger and bigger and bigger cobs. And that's engineering, because you're going from something that's small to something that's big, and you're altering the genetic information, but you're altering naturally existing genetic information. You're not adding anything exogenously from the outside in any way. And so eventually you get a corn that is a cob that is what we eat today. And that corn doesn't exist in the wild at all. You're not going to find any cornfield out in some woods someplace. It just doesn't exist. And that corn can't even exist in the wild.
So in terms of making species of plants and animals that have properties that would not normally exist, although you could imagine, we know that genes can swap, and now we're just speeding up this process by a tremendous amount. And I think that the foodstuff that you're getting now are perfectly reasonable and safe, although I think you want to go into this thing with your eyes wide open. And so another reason that you'd want to do genetic engineering for the benefit of humankind is so you understand the principles of it, so you don't do anything strange.
I think genetic engineering affects each of us daily without even realizing it. In particular, the produce that we're eating are in fact more pest resistant, more tolerant to drought because of the guidance that's occurred in the laboratories. Most of the cows that we're currently eating and the milk that we're drinking are actually clones from the original, or I should say leaning towards a clone. It's still not the true clone. And the reason being is that the ability to make an animal produce more milk is a key thing. So a cow typically probably makes between 100 to 300 liters of milk in a year, but with the engineering that's going on behind the scenes, we're actually making cows that have capacities of up to 1,000 to 1,500 liters a year in production, which is just tremendous. And so why is milk so cheap? Genetic engineering may have had a hand in that one.
The reality is that the principles of biology are the same. DNA is DNA is DNA. The genetic code which makes the DNA work is universal. We learned all these things 40 years ago. The processes are the same. What genetic engineering tells us is that there's universal biological principles, and to me genetic engineering is the most organic form of biology, because we're just using nature's rules, and you're just taking DNA from one organism, putting it into the nucleus of another organism or into a cell in the case of bacteria, and just using these natural processes in order to perpetuate this DNA. So in reality, in this day and age, we can take the gene from any organism and stick it into any organism. In fact, we don't isolate genes anymore. We can synthesize them.
Nature does a pretty good job, and just, as a biologist, trying to understand how nature works and work with it is much more attractive to me than throwing things together kind of like a stew in a pot and hoping that it comes out okay.
Okay, I'll bite on that. Okay, so to create a new species of animal, if I could make an admixture of a human with an eagle, I would do that, because that would certainly solve the traffic problems in Los Angeles. And so, I think that does, even humor aside, I think that does answer your question. If human beings could fly, that would be a marvelous thing.
If I could cross any two species together, I would have to go back to when I was a kid watching cartoons where they hypothetically mix together a millipede and a chicken, and you end up with a long chicken that has lots of drumsticks. And if you're a drumstick man or person, that's definitely the type of mix you'd want to make.
If I could create my own species, I hadn't thought about it, but it would probably be something that looked like Natasha Henstridge.